Hi everyone and welcome. Today we're joined by Dr. Elisabeth Marnik, who is a postdoctoral researcher here at the MDI Biological Laboratory, and she works in the Updike lab. She holds a BS in biochemistry and a PhD in genetics from Tufts. Her dissertation research focused on both genetics and immunology. She looked at how the immune system responds to foreign invaders and makes antibodies normally, and then she looked at genetic and immune cell changes that could disrupt this process and lead to autoimmunity or blood cancers. She's been at MDI Biological Laboratory for 3.5 years. And today Elisabeth is here to talk to us about PCR tech, a tool that is used in both our labs, but also in testing for COVID-19. Thanks for joining us, Elisabeth.
For having me. I'm glad to be talking to everyone today. So before I jump into PCR, which is a technology that a lot of scientists use across the globe, I wanted to remind everybody about DNA. So let me just share my screen. Can you see my screen?
Yes.
Perfect. All right. So most of you probably remember, but DNA is the hereditary information that gives a cell or an organism or an individual all of the information needed to create them. So from DNA, you get all of the proteins that form your muscles, your brain, your eyes, your skin, your hair, etc. But this information contained in the DNA needs to be protected. So it's not used directly to make these protein molecules. It goes through an intermediary called RNA. And then this RNA is used as a template that then is converted into protein. And then these proteins form your muscles again and your skin and all of the things that make up an organism. So I just wanted to remind everybody what DNA is and why it's important. So in PCR technology, PCR stands for polymerase chain reaction. And this is a technique that we use all the time in lab. And what it is when we want to look at a specific region of DNA. So say there's a gene that we're interested in and we want to know if there's a mutation in a specific small region of that gene. We need to then take that region of the DNA and make a bunch of copies of just that specific region so that we can then look and see if there are any changes in that one region that we're interested in. So in this image here on the slide, you have your template DNA that you have designated a specific region that you want to make a lot of copies of. And then what you do is you design these little molecules called primers that bind to the DNA in the region that you want to copy. And scientists have to design these primers every time whenever they're distinct for the specific region of DNA that they want to replicate. So what this works is then after we designed these primers to make copies of that specific region of DNA, it goes through this polymerase chain reaction. And this polymerase chain reaction has 3 main steps. The first step is called denaturation. And DNA is a double helix, so it's 2 strands of DNA together. And to make copies, you have to separate those 2 strands. So denaturation is just exposing the DNA sample to high temperature, and this causes them to separate. And when this separation occurs, that then allows the primers to bind to the DNA where they match. And again, these primers were designed specifically to the small region of the DNA that you want to make copies of. So the next third step is the extension step. So after the primer binds, an enzyme called DNA polymerase needs to bind to that region, and then it starts to make a copy of that DNA molecule. So after that extension happens, initially you may have just had 1 DNA molecule, but by the end of this first cycle, you now have 2 molecules because each individual strand was used as a template to make a new strand. So then this process in PCR repeats anywhere from 25 to 35 times. And at each instance, so for cycle 1, you went from 1 DNA strand to 2 DNA strands. And then you go from 2 DNA strands to 4 DNA strands, etc. So over the whole course of this reaction, you're making a bunch of copies of just that one specific region of the DNA that you're interested in. And by the end of this reaction, you have millions of copies of the specific region that you're interested in studying. So that is the basic techniques for PCR. And that's just, again, making copies of specific regions of the DNA that you're interested in. But there are some modifications that scientists have come up with to allow us to look at this in a different capacity. One is called real-time PCR, which we also sometimes call quantitative PCR. And this just allows us to do that same technique, but it allows us to not only make copies of a DNA, but also determine how much DNA is present. And then there's another version of this called real-time reverse transcription PCR, which is also called RT-PCR. And this is where instead of starting with DNA, you're starting with that RNA molecule. And that RNA molecule is converted back into DNA. And then it allows us to determine the amount of RNA that was originally present in a sample. So these are just modifications that allow us to not only make a copy of a region, but to also quantify how much was there. So at MDIDL, we do this PCR and RT-PCR technology. The most often that thing that I use it for is for what we call genotyping. So oftentimes in my research, I use a genomic engineering technology called CRISPR to delete proteins or regions of proteins that I'm interested in because I want to see what happens when those are gone. Because by studying what happens when they're gone, I can understand what they do normally. And once I engineer a change like that in the genome, I have to verify that that change is being passed on to the progeny of the worms that I'm studying because I use C. elegans. So I use PCR to make a copy of that specific region that I changed to then verify that the change I made is actually there. And then other people at the biolab will use PCR for the same reason, or they may be doing this reverse real-time PCR that I talked about. And they would be doing this because sometimes when you make a change, either a genetic change or you change the environment, this causes changes in the level of RNA that's made from a gene. And sometimes we want to understand how that level is changing. Sometimes RNA levels will go up, sometimes they'll go down. So by doing this technique, we can quantify how the RNA levels are changing from genes that we're specifically interested in. So in the case of COVID-19 testing, so COVID-19 is the name of the disease, and it's caused by a virus called SARS-CoV-2. And this is an RNA virus. So it is a virus that is enclosed in a lipid, which is just another name for fat outer layer. And then inside of it is RNA. So viruses are not capable of reproducing on their own. They have to hijack a host and use the host's own cells to make copies of itself. So when an individual gets infected with SARS-CoV-2, SARS-CoV-2 inserts itself into an individual's cells and then essentially turns that person into a viral factory to make more copies of itself. So in this virus, the genetic material, instead of being DNA like it is in humans, it's instead RNA. And that will be important for something that I'll mention again in a second. So how does COVID-19 testing work? So first, if a patient starts having symptoms, which are most commonly fever, cough, and shortness of breath, they would call their provider, and their provider will decide whether or not it's appropriate for them to be tested. And the requirements for testing vary based on state, so you would need to call your provider to find out that information. But if a patient is approved for testing by their provider, a provider will then obtain a sample from this patient. And this is usually both a nasal and a throat swab. So a swab is put back into your nose to go way back into your nasal cavity and into your throat, because those are the cells that this virus is most likely replicating in if you are infected. And then this sample has to be transferred to an FDA-approved clinical laboratory. And once it gets to that approved laboratory, RNA is extracted from the sample, and then it's combined with all the chemicals needed to do this reverse RT-PCR technology. And this happens in a specific machine that runs this reaction. So I want to explain to you a little bit what happens in this machine. So once the sample gets to a clinical laboratory, the technicians working on this will first isolate the RNA from the sample. And this requires specific chemicals that will extract the RNA from the human tissue sample. And then if viral RNA is present, it will be converted to DNA through some chemicals that do this reaction. And then if that RNA was present and the DNA is now there, you'll have, just like in normal PCR, you'll have primers that bind. And these primers were made specifically to bind to the COVID-19 sequence. But the different thing with this part of the technology is that there's also a probe, and this probe was designed to bind to the COVID-19 DNA as well. And it has on one end a fluorescent molecule and on the other end a quencher molecule. So when the probe is bound to the DNA, nothing's happening. But once the polymerase extends and makes a new strand of DNA, the polymerase knocks off this probe. And then this probe is ultimately cut up. And when that happens, this fluorescent molecule is released and that fluorescent molecule lights up. And then there's a camera in this machine that's doing this reaction that detects every time a fluorescent molecule lights up. So every time a DNA strand is being made, this probe gets knocked off and it lights up. So at the end of this reaction, what will happen is, if you're looking at the machine output, if a patient is positive for COVID-19, you'll see a curve here that's indicated as a positive, and this curve just indicates the intensity of that fluorescence that the machine is detecting every time a probe gets knocked off. And then if a patient is negative, there'll be nothing there, and that will show that the patient either did not have an infection with SARS-CoV-2, or perhaps something technical went wrong and it was a false negative.
Elisabeth, can I just ask, how common is it to have a false negative in a test like this?
So the exact numbers for COVID-19 testing in general are hard to know, hard to find out. I have read that they're estimating that it could be like 20 to 30% of cases, which can be fairly high. But I think we really don't know that information yet, because we would have to have a better idea of how many people have been infected, and maybe because we don't know yet how widespread it is because of testing. So we really don't know how many false negatives are out there. But in general, you can have, because if one of the providers doing the nasal swab, if it's not done correctly to get enough cells, sometimes there's not enough RNA material present to then have this reaction occur normally. And like anything, sometimes things can go wrong in the process. If a chemical was old or something, you can occasionally have. But the laboratories doing this should be making sure, quality control to make sure that isn't happening. But false negatives definitely can happen.
Okay, thank you.
So some of you guys may have heard about this new rapid COVID-19 testing that Abbott has come up with. And this is going to be pretty revolutionary, because the traditional testing can take, the test itself may only take 2 or 3 hours, but because it's so time intensive, because it requires a technician to isolate the RNA from the sample before then doing their reaction, there is quite a backlog. So in some cases, it can take 24 hours or 7 days to get your test results back. But this test is really fast. It can be done within 5 to 15 minutes. And it's very similar to what we just talked about. So first, a sample is collected from a patient, but instead of having to be sent somewhere else, this is being done right at the site of testing, which is great. So a sample, instead of having to be extracted on its own, is inserted into this machine that Abbott has created. And this is a machine that's been around for a while, because providers already use this type of machine for things like rapid flu tests and rapid strep tests. So a lot of hospitals and providers will already have this system in place. Abbott has just been able to modify this system to also test for COVID-19. So after a sample is collected, it's inserted directly into this ID Now machine. And there are chemicals in this machine that break open the virus. Now, because it's a proprietary system, we don't know exactly how this works. We just know the basics. So first, the chemical breaks open the virus to release its RNA. And then there are reagents here that bind and amplify a unique portion of that viral RNA. And just like in the original test, this amplification incorporates a fluorescent marker. And that fluorescent marker is what the machine is detecting. So within 5 minutes, this machine can tell if that fluorescent marker is being incorporated. And then in 13 minutes, if nothing happens, it determines that it's completely negative. So instead of having to wait 24 hours to 7 days, as this becomes more widespread, patients could know within 5 minutes that they have COVID-19. And this would be great because until a positive or negative test comes back, providers have to operate on the assumption that a patient that they're testing does have COVID-19, which means that they have to have all this protective equipment before going in and dealing with them. But if we know that they weren't COVID-19 positive, we can then save all that equipment since we have that in short supply right now, and not have to waste equipment on patients who don't actually have COVID-19. So that was really exciting. And the test is actually the reagents for this test are being made right in Maine, in Scarborough. So scientists in general can do this technology. So we do PCR, we do RT-PCR here at the biolab, but most science laboratories that work with basic research and animal models do not have the government approval to do clinical testing. So the government strictly regulates who can handle human samples for testing. And that's because they have specific guidelines to make sure that both the testing is being done in a way that's accurate and that the people doing the testing are going to be safe, because working with a human sample is exposing those individuals to these pathogens. So to get approval to do human testing for diagnostics, you have to get approval from both the FDA and the CDC, as well as from the Medicaid system. And then you have to get this certification called CLIA certification. And this just makes sure that the lab that's doing the testing is adhering to very strict standards for clinical testing. So even though laboratories like MDIDL have the technology to do this sort of testing, we do not have the government approval to do this kind of testing. And that approval is very hard to get and is not something that you can do unless you have a true clinical laboratory. And the equipment that clinical laboratories use are often different than the equipment that a basic laboratory uses, because they're made for clinical testing. So right now you may have heard that we have a lot of issues going on with testing, and the issues that public health departments are facing are mostly due to shortages. Because there are so many COVID-19 patients globally, all of these reagents needed to do the testing are being demanded not just by the US, but by the whole world. So there are shortages of the swabs needed to collect the sample from patients, because it's not just a Q-tip, it's a very specific swab that goes all the way back into the end of the nasal cavity. So you have to have a very specific swab for that. There's also a shortage of chemicals needed to extract the RNA from the samples, a shortage of chemicals needed to run the reactions, and then most importantly, there's also a shortage of the protective equipment that is keeping the provider safe who are collecting the samples. And then because there are so many people who need to be tested, there is now a backlog of samples, because there's only so many labs that have the certification to be able to do this, and there's only so many staff that can be doing this at one time. But hopefully, again, as this Abbott testing gets to become more widespread, this will help negate a lot of these issues, because it can be done right at the site of testing and not have to go out to another laboratory. So MDIDL can't do the testing because we're not a clinical laboratory, but we are doing things to try to help in this crisis. So we are offering equipment, supplies, and technical help if the state wants it, even though we can't do the testing ourselves. The administration at MDIDL, so Dr. Haller, are in contact with our medical providers here on the island routinely to make sure that they know how we can help and that we are informed, so that if we can offer them some services, we can do that. We are also, individuals at the lab are also working to use our 3D printer to make face shields for our medical providers to help with that shortage of protective equipment. And all of the staff at the MDIDL who can are working from home. So Governor Mills did just make an executive order called your Stay Healthy at Home. And this means that if you are not deemed an essential, if you don't work for an essential company, you need to stay home. MDIDL, because we are a research facility, is considered essential. but we are still making sure that the only people who are actually coming into work are the people who have to be there. So most staff who need to work from home are working from home. And if a researcher does need to be in the lab to do an experiment, they have to comply with all of the social distancing regulations. And we're working to stagger people who come, so people come in at different times. So there's not a bunch of people in the lab at the exact same time. And we are also prioritizing only necessary research. So if there's research that we can put off starting until this has passed, we are doing that. And we're only doing the things that absolutely have to be done, that we can't postpone. And then I have signed up for this system, which is called Crowdfight COVID-19. And this is a system where scientists across the globe are signing up to help COVID-19 scientists. So I'm a scientist, but I don't study COVID-19 specifically, but I do have knowledge and skills that may be able to help a scientist who is studying COVID-19, either vaccines or treatments or other things like that. So with this Crowdfight COVID-19 system, scientists across the world can say what areas of technical expertise they have, and then scientists who are working on COVID-19 can say, I need somebody who can do X, Y, Z. And then the system can put people together to match scientists who have skills with scientists who are trying to help solve this COVID-19 crisis. And that's a pretty cool system, and is showing that even though not all scientists study COVID-19, we are trying to help each other to let this end as fast as possible. And then I just wanted to end by reminding you all of things you probably have already heard a bunch of times about how you can help mitigate this COVID-19 crisis. And the most important thing that we all can do is to help flatten this curve, which you've probably heard about a lot on the news and in other media. So the big problem with COVID-19 is that if we don't slow down how many people get it at once, our hospitals will be overwhelmed because we don't have enough hospital beds or ventilators to treat the most sickest patients. And even though some people don't get very sick, we want to make sure that those who do get very sick have the care that they need. So by staying home unless your job is essential, limiting trips outside, keeping your 6 feet of distance, and washing your hands and covering your coughs and sneezes, you can help to flatten this curve so that less people get infected at one time, so our hospitals can keep up and keep treating the people who need it. And then if you do end up getting sick, it's important to know that you need to call your doctor. You shouldn't just walk into an office because you don't want to spread that around. So call your doctor and they'll tell you what to do. Obviously, if it's an emergency, go straight to the hospital, but you do want to try to call first to make sure you're not exposing providers to COVID-19 without warning. And then science and public health initiatives are very important. So if you can support science and support the public health system, we'll be able to overcome this, and then hopefully life will get back to normal soon.
Well, I just want to say thank you, Elisabeth. That was an in-depth but really easily understood explanation of how the technology works, how it relates to COVID-19, and a good reminder of all the things that we can do to help flatten this curve, keep our medical and healthcare staff safe, keep our families safe, keep ourselves safe, like you say, so that maybe life can get back to normal sooner rather than later. I really hope that the new Abbott test is a game changer in helping us understand the true prevalence in our communities, so that we can tackle it as we need to. MDI Biolab is doing its best to share information that's useful to you, information that you want, information that you need. Obviously, we've had to pivot from a lot of face-to-face and in-person events to more digital content. So I'd like to invite you to follow us on Facebook, on Twitter, on YouTube, and hopefully we'll have more of these types of discussions coming shortly for you. So Elisabeth, thanks again.
Yeah, thank you guys. Stay healthy.